Hope my voice is audible. That it's audible. Wait. Today I'll just discuss about few things related to the human genetics and hope that you guys can follow with that. So basically, it's a crucial part every time in different exams. They prefer to take the genetics part. That is kind of easy things to everyone, because it's kind of more suitable answer for you. So for that purpose, students are preferably do that. Normally I am discussing the different topics, it's not specifically for the exams, because I don't like that kind of exams. Any exams I don't like. I believe about the studies and to know about the things and what we can do it. But like in India, it's kind of all elimination process. A lot of students are there, so who can get the opportunity to that? For that kind of things, the exams are getting conducted here. It's kind of elimination of the students. So for that purpose, exams is necessary. I can feel it also. So that's why I'm just helping people to know about the science more, rather than the exams. So how the feeling of that science, that's very essential. So what you can see on that human genetics part, so like in genetics part there are more uh, disease whatever you want to study in your PhD or in rather than your MTech program. So like that is the main thing, because everyone is working about the disease, so it means the human body is essential. So you have to know about the genetics. So what are the main problem for working with the genetics? Like they are in other organisms like peas or rats or guinea pig or different kind of organisms, or you may say the bacteria and all those. For that you get the control mating. But for the human being, it's not possible to made the like control mating. So whatever we get and whatever we see, we have to taste accordingly. So like an individual might be dominant trait or might be homozygous trait or might be the heterozygous trait. So I've just pronounced three terminologies: the dominant trait and the homozygous and the heterozygous. I think you guys all know about that, but reminding that you should know the what are the dominant type. So you think about the Mendelian genetics, where we have already studied the capital T and small t, and their capital T is like tall. It's if their features is getting tall, it all means it's a dominant type. And if their genome is like capital T and capital T, it means both the capital T's are there. That time you can say it's okay, it's the homozygous, or the small t, it's the sortite. So that is also homozygous. And heterozygous like small t capital T or capital T small t, that's kind of heterozygous. Both the allele are different. So it's for being for a human being, it's really very difficult to mating with that kind of homozygous conditions or heterozygous conditions. So it's being very difficult for that human genome. And then the human have their long generation time. Like human reproductive age is normally reached like 10 to 14, or in might some cases is like 16 to 18 years for the legally cases, or 18 and more than the 18, and in some countries like 21 is a mandatory age for the getting reproductive, uh, going to the reproductive life. So then, like, usually it's take a lot of times, and also some boundaries are there, like human cannot reproduce more than 40 and or like 45 or might be 50. So that's the kind of barriers are there, so you cannot get that the long generation, the short generation time, so the study will be more longer. That's the another problem. And another thing, the family size and the generation generally normally small in the human, that the simple genetic ratios we have learned, like 3 is to 1, you can see them in Mendelian genetics, or they like their genotypic ratio is 9 is to 3 is to 3 is to 1. To maintain that, like you have to take like 10 to 15 of the children, then you can, okay, then you can predict our statistics works with that place. That's kind of very difficult, because today is a live session, I mean live, hope that some people might join or might see. For that, I'm thinking about that every day I will just come and live and to discuss about the some of these important topics. Anyone can join with my live. Okay, so like these are the like key obstacles for studying the human genetic condition. So now another key factors is coming, like there are some of these pedigree analysis are there, like there's an important technique for use by the genesis to study about the human inheritance in a pedigree format. So like it's a kind of pictorial representation of that. So in that picture you can going to the one generation to the another generation. That's uh, kind of interesting. So you can just make some outlines of the inheritance, and some of these common symbols are there, and you can just go through with that, like you know about that the females by circles and male represented by the squares. So like you can say like one first generation cross, that means their circle is there and cross with the one round. So circle you know about that, say females, and square means the uh, and the horizontal line between two symbols representing the man and woman indicating for a mating. So children are connected by the vertical line extended below by the parents, like kind of vertical lines are there, and their person who exhibit the trait of interest are represented, fill their circles and square. And you know about if some person get affected, that means that time what will be the symbol if the male is affected. So you already know about the square. Squares are the male. So now if a person get affected with some disease conditions, that time the square will be filled by black. Normally we are using black, so it will kind of filled box, not the empty boxes. Similar kind of trait for the female will be there, that will be the filled circle. And for other cases, like if you don't know about the male and female, exactly we don't know, that time kind of rhombic structure we can use for that cases. So basically we going to very easy with that things for male and female. Then if you want to get to complicated the things are getting more complicated. Yeah. So now, if any obligate carrier or carrier the genes but does not have the trait, no phenotypic changes are there, but they are like carries the gene of that particular disease, that type of conditions, you can see the box, like if for male case, the box is there. In that box, you can see one black dot. That means gene is carrying with that box, but the traits are not shown. That means this can carry with that kind of disease gene or like mutant gene for different kind of diseases. So for case of female, they're simple. That will be like one circle is there, and in between the circle, one dot will be there, and that for the obligate carrier of that gene. Okay, if a asymptotic carrier, that means unaffected at the time but may later exhibit the trait for that kind of conditions. If we talk about the male, like one box already there and you just cut down the box, this is a simple thing. That means they are asymptotic carrier, like later time the disease may appear, like may exhibit with that disease trait. Similar kind of things with the female. And if you want to make some, it's a multiple person, that means the box, in the box, $1 on s type of sign will be there. That means there's a multiple person. If any person is deceased, that time the slash will be there. And if they are kind of family history is unknown, you don't know about their genetics, that time you put a question mark in the top of the square or top of the circle for female. For the male it will be square, in the top of the square one question mark will be there. And then you just draw about that. That's a very easy thing. And if any kind of identical twins are there, that time you just put the tree and make it the triangular. In the branches, first branches, make the triangle. There's a first branch and make the triangle. Yes. So in that condition it will be like identical twins. If non-identical twins, that kind of both the structure will be there and trace it V shape. And if you don't know the twin is like identical or non-identical, that type of case, you put some question mark on that branch. Okay. And if someone adopted some uh, person, that time if they adopted a male, so you know about the sign of a male and you put the third bracket. Yeah, that's kind of adoptions. And if somewhere like a male and a female, they have their both the sign is like a parallel sign, like a question mark of parallel, and at that time of kids that indicate the they can marry each other, like brother sister marry. Consanguinity is shown with that kind of things. Hope you guys are getting all these points, okay, because these are kind of very easy to explain all those things. And you should know about, and then you just cross about all those features and how it is transmitted to the one generation to the another generation, and then the second to third generation, third to fourth generation, you can easily discriminate with that kind of things. You should know about that the minute part. If kind of first affected family members and coming to attention with the geneticist, that that time they written down P. The box will be there, like you for the male cases it will be like, okay, the square is there and the P sign, because this is the first prevent, first affect, P sign with the straight line. So and another two three things I'll just discuss about that, like what is the reciprocal crosses, because yield the equal results. The reciprocal crosses yield results. That's are very simple. And now I'll just discuss about the two three things, like you can say some of these autosomal recessive traits are normally seen with their equal frequency in the both the sexes and the same to the skip the generation. Like if you think about that, the autosomal recessive traits. So now we are talk about the autosomal recessive traits, and then we will discuss about the X-linked recessive traits, and before that the autosomal dominant traits. So now our focus on the autosomal recessive, autosomal dominant, and X chromosome linked sex disorders. That will be like also some of these recessive and dominant. So how we can easily compare with that the what is the autosomal dominant and what is the autosomal recessive. So that's our main motto. So you have to remember one thing: if skip happen, that's recessive. If skip one generation skip, the disease is there, but skip the disease to the another generation, you just go through the recessive. Or if you find that the no skip, don't skip generation, that means it's a dominant trait. Now you have to check on that it's a X-linked base or it's a autosomal. So if it is a X-linked based, that time you have to think about the what is the male offspring, and is it connected to mother? If it is connected to mother, that means affected male, and it's coming from mother. That means X chromosome is responsible, because the female has a double X, one X will go, and for a male only one X is there and another is Y is coming from father. So we don't bother about the Y, we are just bothering about the X chromosome, it's only coming from the mother. So that's why you just think about that is a X link, and then you or the one another simple funda is you know already skipping and non-skipping. If it is a skipping, and if it is a skip that generation, that means it's a recessive. Hope you guys are understand easily. And now we are talking about the what are the major diseases that the major diseases that can very important for that X-linked phase disease, like that's coming from the mother to the child or mother to the boy, like it's so X-linked disease, like there's a color blindness. Number two, that is the complete androgen insensitivity or the Hunter disease, Menkes disease. Menkes disease is a also another name is there, like that is known as a kinky hair syndrome. So in that syndrome, you find that that's coming from this mother, and it's a autosomal like X-linked recessive traits, and along with some Bruton's disease or like Wiskott-Aldrich syndrome, and the inheritance nephrogenic diabetes insipidus. So like nephrogenic diabetes insipidus also it is inherited. Now we are talking about the X-linked dominant disease. So firstly, we already know the dominant disease, it means in your diagram, or like in pedigree T, or you can say your uh, pedigrees. So in your pedigree you find that the disease is continuously happening. It means it's like do not skip. First thing you have to remember, do not skip. That means it's a X-linked dominant. X-linked. It's a X-linked. Already you got that. Okay, it's a dominant. Don't skip. Now you think about that the mother and child relations. Mother and its son's relations. you find that they already from a father to mother, father to her daughter is there already in daughter too, in every male body, every male child get affected. So that means X-linked dominant. So hope that you guys can understand with that. And in every exam, or you can also predict the some of this pedigree of, you can also make some pedigree of your ancestors if had that some medical history is there in some diseases, you can find it. Yeah, that's a very very interesting things. Now you think what are the important key diseases that related to the X-linked dominant traits. X-linked dominant trait is means male child must be affected. So that is are the Rett syndrome or incontinita pigmenti or like Gold syndrome or Aicardi syndrome. So these are kind of X-linked dominant trait. Now we are talking about the Y-linked traits. That means that can transfer to the father to the male child. That's you have to think about. Now, like Y gene is a very small gene, so it's don't have like more, not more than 50 genes are there. So that's why very less disease are associated with the Y link kind of disease. So it's their role for their differentiation process in the male and the female. So but still we got some of these disease, like some of the interleukins. You talk about their some of the cases, you can find that these are Y chromosome is getting disturbed. So they are already the very low amount of people are getting affected with that. Okay, the passing from the father to son only. Okay, now I think I discuss about all the traits in very quickly. So what you can see easily, like just make some summary on that disease. Firstly, the X-linked, Y-linked, and the autosomal. There you know about the 46 chromosomes are there, like X-linked, Y-linked, and chromosomes. And you already you know about the like autosomal dominant and autosomal recessive, X-linked dominant and X-linked recessive. So X-linked, so and also you know about that the skip and non-skip. It's a dominant, no skip. It's a recessive, it will be skip. That's a very simple thing, and you just think in the exam center and hope that you got the answer keys. Now we talk about some problems like the twin studies. So another interesting, because in my master's thesis I have once time I have done with the monozygotic twin. So like when you analyze the genetics, that means the monozygotic twin and the and the uh, sometime you go for the dizygotic also, like now our concentration will be in the monozygotic twins, like they are coming from a one zygote. If in one zygote their genetics will be the same, exactly same will be their genetics, that will be from the twin studies. So basically to understand the more about genetic changes, like and how environment can influences their genetics, and for that kind of study like epigenetic studies, or like some of these carcinogenic effects of different molecule, and how it can be hampered to the body. So for that purposes people are directly concentrated about the monozygotic twins, like you want to know the exactly what are the major conditions, and for those conditions the changes are happen in the genetics. So like for that cases monozygotic twins, because their genetic makeup is same, So you, for the epigenetic modification, if any kind of epigenetic changes are there in one person and the disease is coming to that, and another twin don't have that disease because their environmental conditions are not the same, or some they are not exposed to the same chemical, that means only these chemicals can modify this kind of genetics or genes, like some methylations or some acetylations happen in that particular position of the gene. For that purpose the disease will come. So for genetic study, the twin study is another key important factor, and people try to find. So in my genetics discussion part, I have just discussed already the obstacles part, the pedigree analysis part, and the twin study. Already I have done in my master's time the monozygotic twin and for fMRI image analysis at that time. And now so you can think about some of the karyotypes. Like, what is the karyotype? Their complete set of chromosome of an organism possesses the karyotype. Like, you can know about the human. Human has the 46 chromosome. So if the band of every chromosomes or the length of every chromosomes is different, but in karyotype you can just make all the, you can use some gene stain and making all those uh, genes, all those chromosomes in a same thing, and you just in a same uh, you can say plate, and you can see every chromosomes, like chromosome 1, 2, 3, 4, and you just compare with the diagram, or you can keep the some standard part, and you just compare with your chromosomes, and whatever you staining, after staining you can find, and making all the chromosomes in a simple tray or in a simple diagram. That's kind of karyotype. So in human karyotype, any questions is coming, that's not a kind of childish question, like 44 chromosomes are there and along with the X and Y are there. So for that they are G banding, and somehow some of these person might sometimes it will be like R banding, sometimes it will be the C banding. Like in different chemical it will be different. Like you can see if GIMSA is used, the special dye GIMSA, that's a G band, and which distinguishes the area of the DNA reach of the adenine and thymine. You just remember adenine and thymine. You know about the adenine bind with the A double bond T. So in the GIMSA stain, the AT bond, the base pair are seen, distinguished. You can distinguish. If you Q bending are revealed the staining of the chromosome with the quinacrine mustard and viewing the chromosome under the UV light, and some other techniques are like you want to know about that the heterochromatin part, like centromeric heterochromatin part of the DNA. For that purpose you can check about the C bending, and you can see the Q bands, and which is a reach of the guanine and cytosine for the base pair, and for that purpose you know the Q banding. So that kind of information type question in some exams, and for that time you can see the Q banding GC, Q, GC, and G for goat, G is for the AT, goat, you just remember the goat. So G, O, AT, AT you know it, know that, and G the GIMSA. So AT GIMSA and Q GC, guanine and cytosine base pair detected by the Q band. So for exam purpose you have to recall all those things. So in major sections whatever I discussed, the three important questions, like in from pedigree part you just autosomal dominant, autosomal recessive, and X-linked dominant, X-linked recessive, and the Y-link diseases, and then you find the karyotyping part. In karyotyping, the AT binding found by the G strain or G banding, like GIMSA strain, and Q banding is reached with the guanine and cytosine or G system. And also you find that the Q bands, like you can staining about the some chromosomes, their quinine masters are viewing by the UV light. That time is a q bending. And also other techniques that you, if you want to know about the centromeric heterochromatin, like euchromatin and heterochromatin, and you want to find the centromeric heterochromatin for a particular chromosome, for that time you have to check about the c bending. So all fours you can kind of getting puzzled, but you have to remember about the two things and others must be remembered. So and then some of these most of the questions will be like what will be the x linked and what will be the y linked, and diseases you can cover. Hope that I think I don't prefer about some recalling things. So for that purpose, but you just think about some color blindness or androgen insensitivity or diabetes insipidus. So nephrogenic diabetes insipidus, it means the some difficulties in the ADH gene. So like, and also the Wiskott-Aldrich syndrome and their Menkes disease. So for that kind of question, so you just think about the X-linked recessive disease, and if you think about the X-linked dominant disease, their Rett syndrome, RETT Rett syndrome, and incontinenia pigmenti, and their Gloth syndrome or Aicardi syndrome. Bas, that will be fine. Now I think in, if you think about all those things, and then it will be very easy for discussing the uh, human genetics. Okay, let's give some questions about that. So how you can concentrate about all these things, like on pedigree it's shown that the female is get affected, female is affected, male, sorry, I changed the question, like male is affected and female is like normally person, they get married and their child, like in first child, like one female is there, so it's completely fine, but male have some problem, and some male female both are fine. So for that purpose, like in generation 2, what is the probability of the child from the parents number 2 and 3? 3 is like fine. First thing in that pedigree, so like there is no some in every generation no skip, so it means dominant, you know about that. So the chance of the diseases, and if had the fourth child, that like they are 1 by 4 will be there when the both the parents and carriers will be there, and the probability will be there, like it's a, so 1 by 4 will be there, and some, okay, let's do some calculation, like, so generation first to second and third to fourth, so we have to know the second generation, third child and fourth child. So third and being and fourth, the heterogeneous, the both the first child having diseases, so like 1 by third cross 1 by fourth, and total probably 1, so that will be 1 by 12. That kind of questions are coming in the exam. If I have some board work today, I want just coming the discuss some type of things, so that's why I'm not doing some board work. So you just have to remind all those things. And some other type of diseases are like the questions are there, the sister shows the trait and her father is definitely the capital A small a, that means his mother is also carrier too, since their second generation does not show any trait, so that means that is, you have to just remember on that. And that's the second 2 has a 2 by third chance of the 1 by. So for that kind of questions, you have to remind all those things, like what are the probability of the first child will have disease and they are being heterozygous. So for genetic things, you always know 2 9 9 is to, so you know about the total 9 character will be there. So what are the heterozygous, like 4 by 9 into the first chance of disease 1 by 4, so you just multiply with 4 by 9 cross 1 by 4. So it will be like 4 by 36. It will be 1 by 9. So that kind of questions are coming. So one day I will just discuss about all pedigree questions along with the whiteboard. So today my target will be like what are the major questions? Like 3 or 4 questions are coming in every exams. It might be CSIR net, or it might be the SED exam, or might be the GATE exam. 2 3 types questions. Like one questions from monozygotic and twins related, and sometimes it's not coming, and but the pedigree analysis is definitely coming. So you know about all those things. I hope that my live will, after my in live end, you can just find all those things in systematically, like you can rehear that, and how the pedigree analysis, also you can make some notes on that. How the male female some person is containing that gene but not still affected, or if it is affected and might have the some other affected, that kind of arrow lines you have to just remember all those things, and then think about that. So these are the kind of things in human genetics. Hope that you guys watch my life, and hope you guys get benefited. So in that life I want to show some of these, how the newly research is coming. So my purpose is there to know about some new research. Hope that my next life I will make some new research, whatever research are coming in that area, we can discuss, or we can just go through some of these portals and see what are the new things coming, or also how to read the research paper. I'll just go through that after that live. Okay, bye bye, tata, good night.